The pandemic offers humanity life-saving lessons. But are we listening? Here are some critical decisions that can benefit, even save our future. COVID-19 disrupted our whole planet, but it could have been a lot worse. Did you know that the H5N1 avian flu is up to 24 times more deadly than the COVID-19 virus? Fortunately, that avian flu doesn't spread easily between people. Now listen to this insanity. A few years ago, genetic engineers created an airborne version of that lethal virus. Although it never escaped the laboratory, more than 100 other genetically engineered microbes and viruses have escaped through accidents. So here's lesson number 1. Don't tempt fate by genetically engineering potentially pandemic pathogens. Just don't do it. Perhaps the world finally has the resolve to make this happen. While we're at it, let's address other potentially catastrophic dangers of genetically engineered microbes that we can do something about now. What we're about to share is breathtaking. Experts reported in 1991 that the world was within 2 weeks of a genetically engineered microbe creating a global cataclysm. In truth, no one is absolutely sure what would have happened if the microbe was released as planned. Some try to dismiss this story as sensational, while others insist that we saved our precious ecosystem just in time. We share this not to create more fear, we have enough fear as it is, but rather to learn and implement another urgent lesson that can prevent devastating consequences. Be sure to stay through the end of this short presentation. You'll be relieved to hear the good news. With the best of intentions, scientists genetically engineered bacteria to convert plant matter into alcohol. The plan was to distribute it to farmers so that they could mix it with leftover crop material to create alcohol, which would run their tractors.
It sounded like a great thing because you could now, instead of field burning, you would rake up all of those residues on your field, put it in a bucket, big bucket, on your farm, inoculate this genetically engineered microorganism that produces alcohol, and in about 2 weeks, open the spigot at the bottom of that bucket, and out comes 34 proof alcohol.
Farmers would then take the nutrient-rich sludge at the bottom of the barrel and spread it on their fields.
This bacterium had been tested through the standard toxicity testing and the fungicide, rodenticide, insecticide testing system of the EPA, and it passed with flying colors.
It was fed to ducks, shrimp, and fish with no problem and was fully approved by the EPA.
I had a graduate student that was interested in genetically engineered organisms.
In an experiment that would ultimately be used to help him get his PhD, he mixed the sludge from the genetically engineered bacteria with soil and planted wheat seeds.
My graduate student went into the laboratory of a Saturday morning and went, oh my gosh, a whole bunch of the plants are dead. All of those treatments where the genetically engineered bacterium had been present were dead. They were slime on the surface of the soil, just green mush. It actually was decomposed by the alcohol being produced by the Klebsiella planticola engineered to make alcohol.
Thank goodness the graduate student decided to do this experiment. If he hadn't, the GMO bacteria was scheduled to be released 2 weeks later.
So we were within 2 weeks of that genetically engineered organism being released out into the field in a trial to show how far this genetically engineered organism could move. 2 weeks.
Bacteria doesn't just stay put.
It would be carried by birds, it would be carried by insects, it would be carried by people, by cars, by machinery, by snakes and voles and shrews and mice.
After this experiment, Dr. Ingham was told in private by an EPA employee just how far the bacteria would spread. The EPA conducted a secret experiment, which they deny they did to this day, where they allegedly released a genetically engineered bacteria in a field in Louisiana and then measured how far it would spread.
It moved 11 miles in every direction in the first growing season. And the next year they could detect it even further out.
According to Dr. Ingham's sources, the EPA stopped funding the experiment, but an employee on her own continued to monitor and test samples.
And my understanding, talking to other people from the EPA, that genetically engineered bacterium can be found every place on this planet. Every place. How could you get it back?
What if the graduate student hadn't done the experiment and the alcohol-producing bacteria were released 2 weeks later?
It would have affected the whole world. Once we release it, it can't be brought back, especially something like Klebsiella planticola, which is found in the root systems of all plants everywhere on the planet. Every time we test, it's there, it's present. All food plants, trees, grasses, brassicas, anything. And it's now going to start to produce alcohol. What's the effect of alcohol in the root systems of plants? It's to kill them.
If the alcohol-producing bacteria were to outcompete and replace the natural version, we're talking about a real-world potential nightmare.
You would lose corn, beans, peas, trees, grass, just everything that exists in a terrestrial system would be slowly but surely destroyed as this bacterium moved out.
Looking at the ecological effect of a bacterium engineered this way, the logical consequence of releasing this to the real world would be that we would lose terrestrial plants.